Our last presenter of the day is Palak Patel, and he's joining us from the National Institute of Allergy and Infectious Diseases.
Thank you, Adriano. First, thanks to all symposium organizers for providing me with this opportunity. I'm a postdoctoral fellow in Dr. Neeraj Tolia's lab at NIH. I'm going to give a talk on neutralizing and interfering human antibodies, create antigenic diversion in merozoite surface protein 1. Yeah. First, I would like to give a brief introduction on blood-stage malaria vaccine development. A malaria vaccine that targets merozoites or blood-stage parasite would directly prevent parasite infection and protects from severe disease and death. And what we need to develop the broadly protective blood-stage vaccines. So we need the essential and strain-transcending blood-stage vaccine antigens. Sorry for that. I just put the painter here. I don't see. Yeah. And the key epitopes that elicit broadly protective antibody responses and immune evasion mechanisms employed by the parasite to circumvent protection. Here, my work focuses on 19 kilodalton C-terminal fragment of merozoite surface protein 1, which is defined as P19 throughout the presentation. As I'm talking about MSP1, I'm going to give a brief overview on proteolytic processing of MSP1. So prior to invasion, PF sub 1 is responsible for extensive proteolytic processing of majority of merozoite surface protein 1, including MSP1, which is processed from high molecular mass protein to 83 kilodalton, 38, 30, and C-terminal GPI anchored 42 kilodalton fragments. And all these 4 fragments are held together through non-covalent interactions, and this results into a mature and invasive merozoite. At the time of Sison rupture, PF sub 2 is released, which further cleaves this 42 kilodalton fragment into 33 and 19 kilodalton fragment. And during invasion, the peripherally associated proteins are shed at the point of tight junction, and P19 remains on merozoite surface. And that may have role in intraerythrocytic parasite development. So now, as we know that P19 is one of the potent blood stage vaccine candidates, now question is how antibodies specific to P19 protect from severe malaria infection. So 3 mechanisms can be proposed based on growth inhibitory activity assay and proteolytic processing based assay. So first mechanism, where antibody binds to P19 in a way that it blocks the secondary proteolytic processing. And the second mechanism, where antibody binds to P19 in a way that, it results into merozoite agglutination. And in another one, it blocks the interaction between BEN3 dimer and P19. And the third mechanism, where antibody bound to P19 on erythrocyte is internalized and stop the intraerythrocytic parasite development. So let's take a look at the structure of P19. It consists of 2 EGF-like domains colored in blue and green. Disulfide bridges are colored as yellow sticks, which are important for maintaining integrity of these 2 motifs. Now, work initiated with the isolation of MSP1 specific naturally acquired antibodies from human individuals. And for that, the tetramer preparation, B-cell sorting, and antibody sequencing were performed by the people listed below. And I performed steps 4 and 5, where I screened and characterized and did structural and functional studies on identified maps. We purified maps, and purified maps were used for ELISA against P19 and full-length MSP1 as control. And based on ELISA results, we had in total 8 maps that specifically binds to P19. So then I wanted to look at their binding kinetics data. And as a result, we had a whole range of maps starting from 0.66 nanomolar to 300 nanomolar. Now, the question is where these antibodies bind on P19. So to answer that question, I did the epitope binding assay. Epitope binding assay is similar to a competition ELISA, and that was performed using biolayer interferometry. First column represents the saturating maps, and the first row represents competing maps. Values in the table represents percent binding of these competing maps. Now here, the cutoff value is 50%, means values below 50% indicates no binding of this competing map. And thus the conclusion is that the epitopes recognized by these maps are overlapping. At this stage, I thought it would be interesting to evaluate these maps for the neutralizing potential. So we performed growth inhibition assay for that. Growth inhibition assay was done by Kazuto and Abubakar from Carol Long's lab. Surprisingly, we had like out of 8, 1 map which showed potent neutralizing activity, and that is around like 93%. So now let me remind you the epitope binding data and the binding kinetics data, that the whole range of map with diverse binding affinity and the epitopes recognized by these maps are overlapping. Now, question is why there is a huge difference in their neutralizing potential. To answer that question, we selected a few P19 human map complexes. Basically, we selected these 3 complexes because of their diverse binding affinity and diverse neutralizing potential, which I'm going to discuss in upcoming slides. We solved the crystal structure of these 3 complexes, and this table represents the data processing and model statistics. Now I'm going to move on to next slide, but at the end of presentation, if anybody wants to take a look at it, I can come back to this slide. From structural studies, we had pretty exciting results. This first structure represents the P19 photodisc map complex, where photodisc is potent neutralizing map, and which has relatively low binding affinity. The second structure represents P19 photo-C11 Fab complex, where photo-C11 is non-neutralizing map with intermediate binding affinity. And the third structure represents P19 photo-C3 Fab complex, where photo-C3 is highest binding affinity and non-neutralizing map. So at that stage, I thought like I should look for polymorphisms in photodisc epitope. So photodisc epitope contains 2 polymorphisms, and none of these polymorphisms had a major effect on photodisc binding. For example, this first polymorphism that is very rare, that A65K, reduces binding affinity by less than 6-fold. And another polymorphism, L86F, reduces binding affinity by less than 3-fold, which clearly indicates that photodisc epitope is broadly conserved. And then we wanted to explore the cross-neutralizing potential of photodisc map. And this right bottom graph represents the GIA for photodisc map with 3 diverse strains of Plasmodium falciparum, suggesting therapeutic application of this photodisc map. Then I superimposed those 3 P19 human map complexes and previously characterized P19 map complexes, which makes it very clear that the photodisc map recognizes novel epitope on P19. And another observation we made was that 25% of photodisc epitope overlaps with photo-C3 and photo-C11. That overlapping portion is colored in red. Now let's take a look at the title. That high affinity non-neutralizing antibodies against the adjacent or overlapping region like photo-C3 and photo-C11 blocks the effect of neutralizing antibody, that is photodisc map. We thought that as antigenic diversion. Now, first question is, what is antigenic diversion? Antigenic diversion is also a kind of immune evasion mechanism where non-neutralizing antiparasite antibodies block the activity of neutralizing antibodies. And that creates another question, that how these antibodies interplay or interact with each other to modulate the parasite survival. To answer that question, we decided to go for combination GIA. Basically, we mixed the non-neutralizing antibodies photo-C3 and photo-C11 with neutralizing antibody photodisc. So at first, in the presence of like combining photo-C3 with photodisc completely abrogates the ability of photodisc map to neutralize parasite. And similarly, combining photo-C11 with photodisc reduces GIA to a level similar to photo-C11 alone. And this right figure shows the proof of concept of antigenic diversion, where this high affinity interfering human antibodies photo-C3 and photo-C11 blocks the effect of neutralizing antibody photodisc. As a summary, this complex structures provide structural and mechanistic basis for antigenic diversion. And antigenic diversion has been observed in merozoite surface protein 1, and that has been proposed from studies analyzing naturally acquired polycyra. Antigenic diversion may be a generalizable phenomenon applicable to other pathogens that can evade the host immune response by simply exploiting the binding attributes of these human antibodies. And we identified novel potent neutralizing epitope on P19. And solution to overcome antigenic diversion is to go for structure-guided design of immunogens that elicit only neutralizing antibody responses, but not non-neutralizing antibody responses. So that's all I have. And with that, the acknowledgement. First, I would like to thank Neeraj and my lab members for their guidance and support. Thanks to Intramural Research Program of DIR NIAID, 4 points. And thanks to all collaborators and thanks to everyone who contributed to this work. And I would like to thank you all for your attention. Are there any questions?
Really nice talk.
Thank you.
Do you see inhibition at the serum level? So, well, first, I guess, do you know if more antibodies do they target the T6 site or the C11 site?
It's like, yeah. So for serum, like what I have observed is the antibodies bind to other part of the MSP1, but not to this P19. For example, that antibodies bind to a P83 fragment that blocks the binding of neutralizing antibody, which is specific to P19. So it's like kind of interrelated kind of thing. So, and another example where they had the naturally acquired human antibodies and which blocked the binding of this mouse monoclonal antibody. So, I mean, yeah, so it's different part of the MSP1 blocks that creates that effect and it blocks. So does that answer your question? Yeah.
Any additional questions?
Okay. Yeah.
So how would you use this? So the last statement was you can use structure guided design to generate those neutralizing antibodies.
Yeah.
So based on that structure, do you have an hypothesis of how you might induce those?
So for that, like we know that for example, like non-neutralizing epitopes we know, and previously identified epitopes with the help of mouse antibodies and all. So that part of the P19 we know that which creates, which generates non-neutralizing antibody responses. So we can use like different applications, for example, the Rosetta and all. So, I mean, that's the hypothesis right now that we can use and like we can modify it in a way that we can generate the, it's basically immunofocusing that we can direct the immune response toward this neutralizing epitope. So yeah, that's basically the hypothesis, but yeah, we haven't like worked on that. Yeah.
If you immunize with a protective antigen,
yeah,
and the protection is not complete, if gradually you will have dominant epitopes generating antibodies that interfere with the protection, isn't it?
Oh yeah, so it's basically the same kind of phenomena we will have, right? For this, I mean, you mean like for the structure-guided design?
So it's difficult to translate this to the field, isn't it?
Yeah, I mean, yeah, definitely it's difficult, but like through the approach of vaccinology, we may direct the immune response. We can guide the immune response towards this. I mean, it's just like thought, but yeah.
Do you think there's possibly any role for this in autoimmunity phenomena, blocking antibodies?
Sorry, I have like no idea about that.
Neither do I.
Yeah, so I mean, yeah, there might be some. I mean, yeah.
Very nice spot.
Yeah, thank you. So yeah, if there are no more questions, I just want to make a small announcement that like Tolia Lab has two postdoc positions open. So if anybody is interested, they can reach out to Neeraj directly. Yeah, thank you. Thank you all.